arthropods, and I think occasionally we're successful. So I'll get on with that. But you know, as evolutionary biologists, one of the really big things that we like to think about is how does a trait arise? How does a new trait arise? So this is a really big question in evolutionary biology. I think we come at it from a lot of different angles, and I'm going to tell you about some thoughts that I have on this today. Because normally the way we think about, all right, how does a new trait arise? How does an organism get a new adaptation? You know, we think about things like mutations, we think about things like gene duplications, maybe genomic rearrangements. You know, these are some of the bigger ones that we think about, and this is how we teach it too. This is how a new trait arises in the population. But what I'm going to talk about today is that, you know, sometimes, and I think we're starting to see this more and more, that organisms can acquire adaptive traits through a process called horizontal or lateral gene transfer. Right? And we're used to thinking about this in prokaryotes, right? So, you know, if we think about our tree of life and, you know, these nice big branches, in prokaryotes we realize that it's not that simple, right? We don't have bacterial species. They're really hard to do, and that's because bacteria swap genes a lot, right? They're moving them around in their environment, they're picking them up through all these different mechanisms, and so we're kind of comfortable with the tree of life, if we think about it in bacteria, as being more of a web-shaped thing, right? That because of this horizontal gene transfer between organisms, we can't really build these nice clean trees. And more recently, we've started to think about that in eukaryotes. So eukaryotic organisms, particularly, I'm a little bit biased towards arthropods, so I'm going to talk a lot about arthropods today. Picking up genes from other organisms, particularly bacterial and fungal organisms, so having those genes actually in their genomes. And a really good example, kind of one of the ones that we've found and characterized really well, is this bacteria I'm going to talk a fair amount about today, Wolbachia. Right, so Wolbachia is a symbiotic bacteria. I think the estimates are, you know, 75 to 80% of insects have a Wolbachia infection or had a Wolbachia infection at some point in their species, lineage. And kind of really early on, we started to find these Wolbachia genes in the genomes of arthropods, particularly those who had them, had an infection with this Wolbachia symbiont. So in Nasonia wasps are a really classic example. They have a lot of Wolbachia genes. In fact, we have found the entire Wolbachia genome in the genome of an arthropod, right? So they'll put their entire genome. So if we're thinking about where these genes are coming from, where we're getting new traits, well, you know, horizontal gene transfer may be a key for some of our arthropod and eukaryotic species. And for a long time we were like, all right, well those genes are getting in there, but they're not doing anything when they're in there, right? They're pseudogenes, they're not really being expressed, they're not really doing anything. Well, we started to find these cases where horizontally transferred genes are actually providing a selective advantage for their new eukaryotic groups, their new arthropod groups particularly. So I'm really interested in these cases. Right? So what is going on? How are they getting there? Where are they coming from? And that's what I'm going to talk about a lot more today, are these functional genes, these functionally horizontally transferred genes. And a really great example came out of the coffee bearer borer beetle. Yes, I did it. Okay, I'm going to have to do it a couple more times. All right, so the coffee bearer borer beetle lives on coffee, not surprisingly, right? And it's one of the few beetles that lives on coffee. It actually can get around caffeine, so caffeine's really toxic. It gets around caffeine because of a bacterial symbiont that lives in its gut, and that actually detoxifies the caffeine. But if you look at coffee berries, they're really high in carbohydrates. They're about 60% carbohydrates, so they're a really great food source if you can digest it and if you can get around the caffeine, right? So the caffeine taken care of by a bacterial symbiont. Turns out that the carbohydrate digestion is due to this manganese gene that's over here. Manganese isn't found in animals. It's actually a bacterial gene. There's no other arthropods that seem to have it, but this little coffee bearer borer beetle does. It picked it up from a bacterial horizontal gene transfer, and now it quite happily lives on those lives on the coffee berries, and has basically no competition, right? Nothing else lives on there with it. It's just it and its coffee berries. So looking all around the world, you know, it's found in all of the beetles, the coffee bearer borer beetles that live on coffee, but not found in any of its closely related relatives. This one little gene here allowed this arthropod to invade a new ecological niche, which is pretty cool. All right, so another example is carotenoids. So we learned in school that carotenoids are produced by plants and fungus. They're not made by animals. Animals use them. They get them from their diet, right? And they're parts of the, they're what make flamingos pink. They're what makes salmon that really beautiful red color. But in all of these cases, they're getting the carotenoids from their diet. They're getting it from plants. They're getting it from fungus. They're making carotenoids. But it turns out that when things started to get sequenced, right? Genomes started to get sequenced. Carotenoid biosynthesis genes were starting to be found in the genomes of a couple different arthropods. And in particular, the first one was the pea aphid. So you can see the pea aphids are sometimes pink, they're sometimes green. It turns out that the carotenoids are what underlie this polymorphism between the pink and the green, and so that's where they actually have the biosynthesis genes. They're functional in the pink aphids. We also find them in the two-spotted spider mite. Again, this is an arthropod that has both a green and a red polymorphism, and these carotenoids seem to underlie this polymorphism, and it probably has a role, something to do with overwintering in the species. And then a third species that this was found on the carotenoid biosynthesis genes, again, multiple genes for this process of carotenoid biosynthesis, was a group of gall-midges. And so when we look at the phylogenetic tree, it actually is pretty interesting because it suggests 3 independent transfers of these carotenoid biosynthesis genes. So it wasn't that it was transferred into the ancestor of these 3, right? So actually, spider mites are very, very far removed from the aphid and from the gall midge. And so what we see is what really suggests that there are 3 independent transfers. I'll get into that for a minute. And it seems that there's probably actually at least 2 fungal donors, though that's not as clear, because these are fungal genes that are found in these guys. And the reason that I say that 3 independent transfers is because if we were to just, based off of what we know of phylogeny, we would expect the gall midge and the aphid genes to cluster. If this was a common descent thing, right, they would be the sisters, and then our little spider mite, two-spotted spider mite, would be off to the side. That's not what we see here. Our gall midge is actually more basal to the aphid and the spider mite, which cluster together up here. And there's also other things about how the genes actually look, right? So where there's introns and all this other stuff. So how the genes are actually arranged that really suggests that these were independent transfer events, which is pretty interesting if you think about it, right? So we've got these shared horizontally transferred genes that are being transferred at different times. So the question that begs is, why are we seeing particular functional genes being transferred, retained, and potentially utilized in multiple species? Now, I just use carotenoids as an example, but it turns out that if we start to really look closely, we see certain genes that are being transferred multiple times. We see amidase genes seem to be common, like the carotenoids, where we're seeing them in multiple species. We're also seeing amidase, peptidase. This whole group of glycohydrolases seem to be kind of transferred multiple times. We're finding them in multiple different species. So all that's a little bit preliminary, but we're starting to see this pattern. And so that's a really interesting question to me, is what's going on? How are they being transferred? Why are they being maintained? And what's going on? So some ideas, some hypotheses as to what's going on here. Well, it could be that some donors just transfer genes a lot, right? I'm looking at Wolbachia, right? We know that Wolbachia, it's kind of slutty, it gets around. It's getting in all these different genomes. So maybe it's just, we're just seeing a lot of Wolbachia genes, or there's a couple different donors. There's maybe like a fungal gene, or a fungus, a species of fungus that's really gotten around and really spread its genes. Or maybe we see some recipient species that are more likely to take up donor DNA, right? Some species, maybe aphids, that's actually not the case, so that's actually a bad example. But maybe there's some species out there that are just, they pick up a lot of genes, right? Whatever's in the environment, they're kind of picking them up. And that may be due to lifestyle or their ecology, right? So these aren't mutually exclusive hypotheses, but these are some ideas about kind of answering this question. And the one that I'm a little bit enamored with at this time is that these genes, maybe they're important for ecological niche invasion, or maybe they actually underlie these dietary shifts. So similar to what we saw with the coffee bear or borer beetle, if you pick up this gene, well, now you can have this new lifestyle. You can invade this new ecological niche. So maybe we're seeing these shared horizontally transferred genes, and they're in the same ecological niche, kind of like our carotenoids. Those we're all kind of phloem feeders, right? So maybe that they're more associated with an ecological niche than with a specific donor, or just sort of these general reciprocal. Sort of some species are picking and some aren't, sort of seems to be that right? All right. So to answer this question, the first thing we need to do is identify a lot more horizontally transferred genes in a lot more species. We've only got kind of a handful going on. And this is where I'm spending the bulk of my time right now. And we've kind of come up with a three-pronged approach. I'm going to just generally tell you about them now. Right now, method one, kind of follow the bacterial identification pipeline. We'll go a little bit faster. But basically, we're taking our target genome, we're looking at, we're blasting it through a database of bacterial genomes. We're looking at comparing that to a database of animal genomes, and we can identify genes with significant bacterial identity, low animal identity, and go, these are bacterial genes in animals. The other one is we're looking at a niche-specific horizontal gene pipeline. So we're taking a target genome, we're looking at closely non-related, non-niche sharing species. Anything that doesn't hit, we're moving in, looking into another group of distantly related niche sharing species. So we're looking for genes that are present in a distant species and not the other. The third one is kind of a brute force. We're going through the literature, we're finding previously identified horizontal genes, transferred genes, and we're looking for them in new species, so in new places. And right now, we're sort of using the kissing bug, Rodnius prolixus, as our current focal species to do this with. This is kind of one of our first trees that we've used with the first two pipelines. We've identified an anchorin repeat gene, which isn't too surprising. We found this in a couple other species before. But what you can see is that these are mostly Wolbachia in blue. Again, Wolbachia shows up. But we found that this gene is shared in multiple species of arthropod. Here's our Rodnius gene down here. So this wasn't identified previously as being in Rodnius, but we're seeing these anchorin repeat genes repeatedly shared through multiple independent transfers, it looks like, when we start to build the trees up. So this is sort of our preliminary run at it. Some very other preliminary results. We're seeing lots of Wolbachia transfers. We've actually identified several peptidases. Those trees aren't quite ready for prime time, so I didn't bring them out today. But we've also identified several horizontal chain transfers that were previously identified in the genome paper, which means that these databases, or these pipelines that we've designed, are working. And we've got some more candidates, so it's kind of a stay tuned. We're going to keep building trees, looking at some other stuff, running some other species as well, to sort of answer this question of the multiply horizontally transferred genes. So that's where we're at today. I'd like to thank my students, and also if you're interested in teaching and research postdoc, come talk to me. We have those at Spelman. We're a liberal arts college, and we're always looking for interested people. So I think I can take that.